Looks like we narrowed down to the 4 ML people. We're still hanging out, which is awesome. And so the first half of the class was, I'd say, a little bit more basics and background. And I think starting with today's lecture, lecture 5, and moving forward, we're going to dive deeper into, at least for today, today I'll be covering chemical modeling with transformers. And yeah, so this is the fifth lecture in our series, Machine Learning for Biochemical Applications. I'm Aaron Feller. I taught the second class on programming using AI agents. So yeah, so who's heard of a transformer? Most of us, okay. A few of us haven't. So in this course, I'm going to go through an example of chemical modeling sort of with this type of transformer architecture on a really basic level. And then we'll dive deep into that architecture and how it works, into the nitty gritty. So let's get into it. So just to start with a question to set up our example, something sort of abstract. Can we use ML to predict scent? Right, so like in this sort of example, we're taking like a far leap all the way to like human scent receptors and the labels that we give smells, right? Can we go from the chemicals all the way to the way we smell them? So we'll walk through this example. So we know that objects have scent from the presence of specific molecules. There are 5 flowers here. We have rose, carnation, lily, and just the names of different chemicals. So you can see there's 6 here for the rose. There's a little bit of overlap, like linalool occurs in the hyacinth as well. There's some overlap of these chemicals. When we smell that flower, we give it like a certain label. And we know that molecules have specific scents. So different molecules activate different receptors, and we give these smells different labels. So there's a large label data set that can take us from the chemical structure to the scent of that chemical that we perceive. And you can see there's sort of groups here like floral and green. You can see some similarity in these fruity esters. Also, there's like a ketone in all of these groups. So there's some structural similarity here, but it's not always indicative of how the molecules smell. So this is just a simple example from a paper that came out this year in August, where we have an anchor molecule here, isoamyl acetate, which has a specific scent profile, sweet, fruity, solvent-like. And this actually comes from the fruit, the banana. We take a molecule with a very similar structure. We change 1 carbon to a nitrogen. And we find that this molecule smells very different. We have something that smells fishy and animal-like, right? So if I'm lighting a candle in my house, we vary the chemical structure a little bit to make a new scent. I'm not trying to get a fishy animal smell. It's usually what I'm trying to hide when I light a candle, right? But then this molecule on the right side, very dissimilar structure, has an aromatic group. And this scent profile here, alcoholic, sweet, and fruity, matches to our initial anchor molecule. So doing a basic structural analysis on scent isn't necessarily going to work. So we'll walk through how to use machine learning to solve this problem. So jump right into some of the architecture. Transformers can convert text to numerical vectors. So at the bottom here, we have language split into words. We pass it through this transformer network, which we'll get into the structure of this later, and we get out a feature vector. Luckily, chemicals have also been converted into strings. We can take a chemical structure here and follow the longest path through the molecule, build side chains off of that path, and then just label the atoms on each of these paths. So starting here, we have an N number 1. You can see it's the first atom here. We move through 2 carbons to another nitrogen. And then we get this branch. So we open a parentheses, have another 2 carbons with another number 1, return to the original path, and so on. We can go from chemical structure to a one-dimensional text string. So once we've done this, we can take that text string, split it into tokens, and pass it through the same architecture, produce a feature vector. Okay, so what do we do with the outputs of this? So what do we do with the feature vectors? So going from the top, we have chemical inputs. We pass it through this transformer model to conduct our pre-training task. We pre-train this model. Then once this is complete, we can pull out the feature vector, the embeddings from the last layer, and apply it to a new model called transfer learning. And then this new model can predict scent, right? Or predict the labels that should correspond with that molecule. So out of this, we can do PCA analysis, principal component analysis. And so this first figure here is the true labels of the molecules. So the principal component 1 and principal component 2 on a 2D plot here. And we can see with the true labels, we pull out the ethereal scents here in green, floral scents in purple, and umami or meaty scents in red. And they're well separated. But if we do the same thing with fingerprints or a structural type of analysis, you can see all the scent labels are overlapping, right? It's not very good at separating the different types of scent. When we use machine learning to produce these feature vectors, run PCA analysis, and you can see, again, good separation between the 3 different scent profiles. Yes.
The true labels.
That's a great question. I don't know the specific. Yeah. Yeah, so there's a way, I pulled this slide out, but there's a way of representing molecules where you pull each kind of like functional group or section of the molecule apart, and then you build a long vector that's a one-hot encoding of the presence or absence. Correct. Right, so this is true labels, a structural based analysis, and this is the machine learning base. Yeah. Right. Yeah. Right. Okay, so we see the separation with PCA on these features. And then I just threw up the actual result. So this is a head-to-head with humans. So all the gray, let's see, let's go through this. So x-axis is success or correlation to the panel mean, or how close the prediction was. And then the y-axis is all the different scent types that the model attempted to predict. And so the orange dot's the GNN, the neural network that they use in this case. And all the gray values are a panel of human beings who sniffed the object and made a guess at what it smells like. And so the data here sort of shows a correlation between how well the model is and how well the human is doing. And then just a note, so essentially the success here is not perfect, right? We can't grab a molecule and predict scent perfectly with this model. And there's a few caveats here. So in the paper, they did GCMS analysis, and they just grabbed 50 compounds from their total study. And of those 50, they found only 11 of the sample odors were produced solely by the nominal compound or the compound that they thought they purchased. There's a lot of contamination in the data set. The other 39 out of 50 contain contaminants contributing to scent in a minor, major, complete way. Yet despite this, the model achieved scores higher than the human median, right? Despite all the noise in the data. It's a second model that they trained. Yeah. Okay. I'm not positive. I didn't dig too deep into the paper and the data set, but I mean, sort of just as an example to show that we can use machine learning in this way. And that the data set was pretty rough. They tried to clean it up, but we still like found a signal through the noise. The graph neural net. So I know we're talking about language, but graph neural nets use the same architecture. So in this example, it's a similar architecture. All right, any other questions? Okay, so now I'm going to jump into some of the behind the scenes of how the model works and get into like detailed steps through a transformer model. Right, so how do transformers work? We've all heard of the problem of explainability in machine learning, right? So we have some sort of input like the chemical structure, and we're outputting the scent profile. And what happens in this black box, I mean, sort of a joke, but you know, input is converted to output magic. We'll dive into this black box and at least explain the steps that the model is taking, even if it's not perfect. So transformers consist of an encoder and a decoder. This is a really basic example of, you know, this is the full transformer model. You have this encoder that takes an input, text, I like science, passes that to the decoder, and we get out German, right? So a simple translation. And I just threw the second figure up there. That's what Devansh had in his example last week. So a similar type of model where in the center here, you get this compressed representation for a feature vector. So to take it a step deeper, this is the full transformer architecture from the initial creation of it. So this paper is called Attention is All You Need. Came out 6 years ago, and this is probably the most reproduced figure in machine learning at this moment, I would say. And so we can split this here again into the encoder portion on the left, split down the middle, and a model built with just this structure is called a BERT, or bidirectional encoder representations from transformers. And then on the right side, the decoder is a GPT, or generative pre-trained transformer. So this is like ChatGPT is built off of this type of architecture. Right. Yeah, so translation problems where you have an input in one text and an in one language and an output in another, you'll encode your English language and you'll decode to German. So the representations that come from the encoder will be passed, right? That sort of like stored knowledge of the meaning and the semantics of the English will get passed into a decoder to go from that feature vector out to spoken German. And then today we're just going to focus on the BERT architecture. So if you notice, before we jump into it, a lot of the features are the same between the two. So multi-head attentions here, feed-forward networks here, we have positional encoding. So we're going to cover BERT, but you'll have all the basics to understand GPT as well. Okay, so this is sort of the layout that we're going to map this system with, and we're going to step through it in 4 steps. So the first step, we'll look at the input embedding. So how do we get from text to an embedding that goes into the model? We'll then look at position encoding. And so this is how the model learns the positioning, or like what order the words go in the sentence. Section 3 will actually look at the attention mechanisms inside the transformer. And then finally, the output embedding, or how we produce a result. So the first step in the input is tokenization. So this is how we go from text to numerical representation. So this is done in just a simple dictionary lookup. So you have a list of characters that occur in your text, and you just assign a single digit to each of those. Right? So in this example, you can see on a character level encoding, you have numbers representing each character. And if a character repeats, we assign the same exact number to it. So all I's will be 3. And so on. S's are 4, right? So you just build a one-to-one relationship with me. We can tokenize on the word level as well. So every time this shows up, it'll have an assignment of 1, or on subwords, right? Token and izing, right? Izing is going to occur more frequently in your corpus. Then once we have these numbers, we have to produce a matrix. Transformer models take input of a matrix of a specific size. So the way that we get this is they're called embedding, word embedding. And we just train a simple linear transformation that goes from these numerical values to a specific token vector. So this linear transformation isn't programmed in, it's learned by the model during the training procedure. Correct. I'm pretty sure it's just a linear transformation. So it takes, let's say you're going from the single digit 0 to 768 digits. It'll just have 768 operations of linear transformation. And so that transformation is unique for each token. So every time a 0 shows up, it'll produce the exact same embedding.
Yeah, but that's not really.
Sure. Correct. Right. Okay. Okay. Sure. Yeah. I think it functions in the same way. So this actually, that occurs second. So we build right. And I think it's called a linear transformation because for back propagation, you need a function
Possible that
to update the weight. Yeah. Right, right. Yeah, so I mean, at the end of training, when your model's fully trained, every token ID of 0 gets the same exact embedding. Yes. So that length is actually, you can set that to whatever you'd like. Yeah. Right. There's more information that can be stored. And like based on the size of this token vector, your like the whole model will shrink and grow by the number of like the matrix multiplication, the tables will grow. Right. Okay, so this is, yeah, that's, go ahead. So from converting from the token ID to the token vector, that's learned in the training process. So it starts randomized. It'll produce, completely random, but constant for each token input, and then it'll update over time, model training. Okay, so that's how you generate the input embedding. The next step is position encoding. So positional encoding is just a way for the model to store the position along a sequence. So anytime we're using like a 1D kind of linear chain, like English, like text is read, or chemical structures built, or sorry, our text representation of chemical structure is like linear format where the order is really important. If you have no way to encode that order, then you could scramble the entire sentence or the entire chemical and you get the same exact result. So the way that position is encoded in this way is just two simple functions using sine and cosine. So and with those two separate ways, we're able to produce a graph that looks like this. So this gets produced through, you take your sequence, and it doesn't matter what these tokens are, they could be anything. And you grab your index, so 0 through however long your sequence is. Then you build out a matrix of your hidden dimension for that 768 or whatever that value we chose for this, the same size for the embeddings for the token. Right, so we end up with the same size matrix where we perform sine and cosine operations. We get values at each cell in that matrix between -1 and 1. Right, so it's just a slight weight. And then these get added cell-wise or pairwise across the entire matrix. So the weights just get updated slightly, or not the weights, but the actual embeddings get adjusted slightly, either up or down. Maximum value of 1. Right. Right. So this just maintains the order so that the model can understand, I comes first. I am a robot, not robot am a I. Although that kind of works. So that's just added.
There's like other
Right. Right, and I guess I should say I'm talking about the initial BERT architecture and the first paper that was published with this sort of encoder. Okay, so then the next section is the intention layers. This one's a little bit complicated, probably the most complicated portion. So the first step in this, and we'll explain what these 3 values mean, but the first step is to generate a query, a key, and a value. And so we take our embedding vector that we produced with positional encoding at each position or at each token. And we do 3 separate linear transformations to produce vectors of the same exact size, but now different. We're splitting this vector, we're transmuting it 3 different ways. Produce 3 vectors of identical size that contain different values. And again, with this linear transformation, everything's learned as well as the model trained. Then the query key values are used as a search and retrieval system. So each position has a query and a key. And to propagate that position forward, you take that position's query, multiply it by the keys of every other position in the sequence. And this produces a single value, almost like a weight. So the closer they match, the closer this value is to 1, and the more different it is closer to 0. And you multiply this scaled between 0 and 1 by the values that correspond to the paired keys to get a single output value. Right. So you're essentially, each position has a value associated with it, and you're choosing through here, through the search, or this multiplication of key and query, how much of that numerical representation you want to include for your next step. So this is a breakdown of the matrix multiplication really simply. So you take your query times your key, and you produce a heat map where there's numbers scale between 0 and 1. You then multiply that by your values to get Z, or your output. So everything we just talked about fits into this box here. So you have your query key and value, your matrix multiplication. There's some scaling operations in softmax to fit it all between 0 and 1. And then again, matrix multiplication against your value. And so this is a single attention head. And we actually run these in parallel. We do this operation, however many heads you want to set, the value H. Right. So
What does it do?
I guess it's called attention because it allows like the output of a single neuron to focus on the input from various elements. Right. So if you have a sentence, the cat walked down the street. Trying to figure out the meaning of walked depends on having a cat to do it. And it depends on, a down the street in some sort of way. The cat doesn't eat down the street or sleep down the street, right? So we have to take in context of specific words in different positions in the sentence. So allowing us to add weights and incorporate values from specific locations through a text string is sort of the result of it.
Text about like where parts are more relevant to words. Like which
Right. Yes. That's a good question. So everything's randomly initialized with a value between -1 and 1. It's actually generally between -0.1 and 0.1. So a small variance. The way I like to think about it is based on your architecture, it will learn a representation that you've sort of directed it to. So by doing the matrix math in such a way where the query and key are multiplied first and softmax at the end, it sort of pushes the training to allow it to run like a lookup table. But yeah, there's initially at the very beginning, completely randomized. Correct. Right. Yeah, I would say there's probably thousands of lectures on this topic. And like scouring through them, there's so many people attempting to understand this. And it's quite difficult, I would say. So I've tried to sum it up as best as possible for everyone here. But yeah, there's plenty of, I didn't build this, I pulled it from a blog. Right. Sure. Sure. So softmax just scales all the values so that the sum is equal to 1. Right.
Softmax values.
Right. You can think that like the propagation at each position is some weighted sum of all the other values in the entire network. Right. Okay, so that's one attention head that gets put into multiple heads with multiple nodes inside of it. And essentially the values that come out get concatenated and then, I hesitate to say, linearly transformed into a vector of the same size output as your input. So we're taking all the information that was produced by the attention mechanism and then shrinking it back down to size. So that's attention in a nutshell. I think there's some small pieces I didn't fully explain, but there's a lot of resources out there if you're curious about like the minor other points. So then the final step is we've gone through these transformations. Now, like how do we produce an output embedding and what does that look like? Great, so we have our BERT model here and we just take the last layer, pass it into a feed-forward neural network or a fully connected layer, and output a vector the same size as our dictionary. We do a softmax over that, so it's a probability distribution of what word in your dictionary or what position in your dictionary the model thinks should be in that place. Right, so in this example, you can see we're going from AA aardvark to ZY Z Ziva, both close to 0%, but improvisation in this case is the word that the model predicts. And this, again, the softmax function is what makes it scale the percentages. So this is actually how we train the model. So we can use the loss function that pushes the answers closer to the ground truth. So we take a sentence that we know the input of, the cat is eating some food. We mask a certain position, in this case, the word eating. We pass the masked sentence through our model and just look at the output at this position. Then our loss function will push the results or the answer toward the correct one because we have the ground truth right here. Any questions? This here? Yeah, so I didn't cover this. It's just a skip connection. So it actually takes the output from the transformer head and adds that pointwise to the previous layer's output. A handful of reasonings for this, but the main one is that so it doesn't forget over time as it passes through layers. You're actually sort of maintaining the previous layer. So that change occurs slowly over multiple layers of the transformer. Okay, so I guess with this, what we can now do, we have a pre-trained model. We can cut off the language modeling head is what it's called, or the portion that predicts the vocabulary. We can stick any type of model on the end of this if you want. We can add a linear model or add a few deep layers or do a random forest on the end to then predict any output value we're interested in. So this is, I guess, the transfer learning portion. So just to give a few examples of downstream applications, so a recent publication from IBM research called Mole Former was trained in this way with chemical language. The pre-training was done on 1.1 billion molecules. And the output of the model, they found that they could do a pretty good job predicting blood-brain barrier permeability, right? So for medical use, drug treatment, and neurological disorders, pretty important. Band gap energies, so in generating molecules for solar panels, band gap energy is the amount of energy that can be produced from photons, as well as antiviral activity, right? So these, once you've pre-trained a model on enough data, there's really a lot of applications. So I think that's all I have. I have a couple extra slides on visualizing attention, which we can go over as well. But if there's any questions. I don't know. I think what they did is they just took off the language modeling head that predicts the dictionary and had the output be a single point, and then fine-tuned across the whole model to update the weight. Mhm. Definitely. So for proteins, we can do amino acid representations, so like a single character for each amino acid. Chemical representations, we can use like individual atoms, so like carbon, nitrogen, oxygen, or open close parentheses. In DNA models, I think Devant covered a little bit of this, but we can use individual nucleotides. Then you only have a dictionary size of 4. You can do dimers, trimers. Mm-hmm. So yeah, 100%. So I'm working on training my own model of this style now on peptide with atomic resolution. And there's something called byte pair encoding, which I'm working to implement, which essentially, so if you use a single, let's say you have something that scours your entire database you're going to train on, finds every individual character, gives it a single token. The skew or the distribution of that is going to be really poor because Like for my example, there's a lot of carbon. There's some nitrogens and oxygens. There's gonna be a lot of like parentheses, right? So you're gonna have most carbon, parentheses, and then you're gonna have a handful of tokens that are minimally used, so the model doesn't see it very often. So byte pair encoding essentially takes the most frequent occurring tokens or characters and finds what token occurs most likely next to it and bins that into a single token. Now you can get like a CC as one token. Right, so it actually increase your dictionary size. You still maintain, exactly. Like if you take a single carbon as one token, double carbon as a second token, you're gonna have an example in your data set where you just have a carbon flanked by oxygen. So you still need a token to represent that character. So in my experience, having a skewed data set where you have a whole bunch more representation of a single token actually hinders your model from learning because it can cheat and just predict everything is carbon. It's like going to the roulette and 90% of your spots are black. You just keep betting black. But potentially, eventually, it'll converge to a meaningful representation, but it takes a lot longer for it to get
That is having more models. A lot of the chemistry I just
there. Bipair encoding, which just looks at frequency. So it's twins, I would say. So you take your highest frequency, whatever amino acid occurs the most, and see what occurs the most next to it, or what twin. Then it iterates, right? So now you have a new token for those two. And if something occurs next to those two as a single token, right? So it's pairing two tokens, not two characters. You set a limit. You can set a vocab size limit, or you can set a limit for like prevalent that you want percentage wise. Every token that has more than this percent, I want to reduce it under a certain occurrence. I mean, not to get, I think in biology, we're likely, at least for explainability, we want to stay on tokenization of a character, an amino acid, or something that makes sense. But in language modeling now, it's done on the bytes. So what a character encodes in bytes in the computer, the byte pair encoding actually occurs on that level. Different languages. Yeah. Oh yeah, so I can show, so attention through the model can be depicted on the molecule. So you can back propagate through the whole model and figure out how much contribution each portion of the molecule was given, during the attention mechanism. And so let's see. So when we select this carbon here, you can see what attention was paid to the previous layer, or the values from the previous layer. Same thing's occurring here. And then we can depict that across the molecule, right? So whatever point in the model we're looking at in this case, these two carbon atoms were important for the decision it made. What's interesting is if you have your model go all the way out to a reported result, let's say you're now predicting blood-brain barrier permeability on like a number scale, you can back propagate from that number and see what it paid attention to for the decision it made of the score it gave. Right. So there's, it was pretty quick, but I'll share a link that has a Google Colab with this preloaded, and you can actually walk through a model and see across, I think it's 5 layers here with 8 heads at each layer, what the attention was focused on. So you can take, this code can be adapted to any model you want to import as well. And that's all I have. I hope I didn't confuse everyone more than when you walked in.